Hello. I'm still missing the presentation by Mariano Gavito. Is he in the audience? No? Okay, we hope he will show up. Hello. Hello, everyone, and welcome to this second day for the HiTSeq. Sorry. We are starting a new session, and before starting, I would like to remind you that we count with the sponsorship of Amazon Web Services this year. This has been a great opportunity for us to bring our keynote speakers, and they will be having a demonstration and a talk, industrial talk, this afternoon after the coffee break. So please stay around. I think they have very interesting things to show us on how they are supporting genomics research and sequencing. So today, we are starting this session with our second keynote. So Andrew Adey is talking today, and let me give an introduction about Andrew. He did his PhD at the Molecular and Cellular Biology program at the University of Washington in the lab of J. Sender. And during his graduate studies, he pioneered the use of novel transposase-based methods for rapid, low-input DNA sequencing library construction. And that technology he later extended to the genome-wide analysis of DNA methylation. He also applied long-range sequencing methods to produce the first haplotype-resolved genome and epigenome of an aneuploid cell line, like it was HeLa, where he investigated the role of haplotype and copy number on the epigenetics and transcriptional landscape. After completing his graduate studies, he started his own independent group at the Department of Molecular and Medical Genetics at the Oregon Health and Science University, and he is now associate professor on single-cell technologies, where he's profiling a variety of epigenetic properties with an emphasis on chromatin accessibility. The Adey lab is currently focusing on the development of single-cell technologies for epigenetic research with an emphasis on chromatin accessibility, and he's applying this to neurodevelopment and cancer biology. His work, the work of his group has also recognized by the American Society of Human Genetics with the Early Career Award in 2018. So where is Andrew? Okay. So he will talk today about single-cell genomics using combinatorial indexing, and we hope that we can get your presentation. Yes. Yeah. Okay, welcome, thank you.
All right, is the microphone working? Yes, okay. Well, thank you for the introduction, and I really want to thank the organizers for inviting me. It's exciting to be here. I'm going to be talking about some of the work that we're doing in my group, really developing technologies to assess single cells for multiple different properties. I'm going to focus on one in particular, and really some of our efforts to really expand throughput and incorporate other types of information. So most studies today really focus on single-cell RNA expression, and there's a number of good reasons for that. First, it's a highly informative mark for cell state. So when you're assessing transcript abundance, you actually get a lot of information out of that, and it's very useful for cell state dynamics, and lineage tracing, things like that. It's relatively easy to access. So mRNA molecules have this really beautiful poly-A tail that it's a nice molecular tag that you can latch onto with a primer and extend into that. So it's a nice way to access those molecules pretty readily. And it's also fairly easy to interpret, relatively anyway, because you're dealing with genes, right? I'm a geneticist, I think, in terms of genes primarily, and we know what most genes do. Certainly, there's a lot of work still to be done there, though. But beyond gene expression, there's a lot of other stuff going on, right? So, you've got the genome itself, and Alexis's talk earlier was talking about how a lot of rare variants can affect gene expression, right, and splicing. You've got DNA methylation, often repressive when it's at promoter regions. There are regulatory proteins, so many different transcription factors, other DNA binding proteins that act on regions of the genome that are these regulatory elements, or regulatory sequences here, where these are locations in the genome where there are no histones bound, there's no nucleosome present. It's open and accessible to the different machinery in the cell to utilize those sequences. As well as dozens of different histone modifications that are activating, repressing, all sorts of different things. And even the three-dimensional structure of chromatin in the nucleus can play an important role. So we really want to look at a lot of these. Today I'll be talking about a few little vignettes. One is just the general platform that we use to assess single cells and different epigenetic properties with a focus on ATAC-seq data. And then a little project that we have in the lab using it to study corticogenesis, and some of the I think, interesting things we can use the data for. A newer project in the group on trying to incorporate spatial information. So this is kind of difficult with the ATAC-seq, but we've got kind of a crude approach, but it works pretty well. And then some future directions, some new things that we've been doing to really expand things out. So first, our platform. So why are we interested in ATAC-seq? I like to use this paper in particular to motivate the use of ATAC-seq. This is from the Greenleaf and Chang labs at Stanford, where what they did was profiled, they're able to sort out cells from blood lineage populations, and you can do this very readily with different cell surface markers. And then they did ATAC-seq and RNA-seq on these samples. And what they were able to show is that when you cluster out the cells based on the ATAC-seq signal, it actually does a better job. It achieves a higher cluster purity, so there's more information separating out these cell types that you can use. And if you think about it, it makes sense in a lot of ways, right? So gene expression is analog. You've got a pretty big dynamic range as well. But there might be fairly subtle differences in the gene expression level between two cell types, right? So that can be a challenge. Whereas regulatory element usage is almost digital. It's almost active or inactive. We do use counts data, but primarily you'll see sites are either present or not. That's a little easier to resolve. But the biggest reason here is that there's over 2 million annotated regulatory elements in the genome and only tens of thousands of genes. So there's just a lot more bits of information that are actually going into this. And furthermore, they broke it down into promoters versus distal elements. And not surprisingly, distal elements works better, but from what I just told you, with only tens of thousands of genes versus over 2 million regulatory elements, there's a couple orders of magnitude more data going into the distal element one, so I think that's one reason. But what is particularly interesting, though, is that when you remove the promoter signal, you're actually getting a higher cluster purity than if you included it with just all the attack-seq peaks. Which indicates that distal elements are far more cell type specific. And this is something that's been known for many years with DNase hypersensitivity data and things like that. Also, the actual promoter accessibility does not correlate well with transcriptional abundance, so that's another important point. So how do we actually access this mark? So we use a transposase-based strategy. So if we take our hyperactive transposase and load it with sequencing adapters, expose it to purified genomic DNA, it'll fragment the DNA and append adapters in a single enzymatic reaction. This is now the Nextera technology. And it'll produce genome sequencing library on the just purified genomic DNA. However, if you apply it to native chromatin, the same concepts that apply to DNase hypersensitivity and MNase apply to the transposase, where it's actually sterically inhibited from acting on DNA that's wrapped around nucleosomes. So it will insert with a very high preference into these regions of open chromatin, and therefore you'll only get viable sequencing molecules from these open regions of chromatin. When you sequence, you get peaks of signal, and you can call those, and those are your open active regulatory elements. So how do we take it to the single cell level? With RNA-seq, there's a lot of different strategies that have now kind of been adapted and moved over to attack-seq. I want to highlight a paper from the Teichmann lab where they did a great job with this technique to do single-cell attack-seq where you have a single nucleus in each well of a plate. This works really well if you have a very low number of cells that you want to profile. In fact, my labs, I think we're doing it where we're tagmenting nuclei and then mouth pipetting them individually into wells of a plate because we just have such a small number of cells. One of the original papers to do single-cell attack-seq was done on the Fluidigm C1 system from the Greenleaf and Chang labs using that platform, though it's quite expensive and also fairly low throughput. Now there are two commercially available droplet-based methods that both work quite well, and they're quite easy to run, so I highly recommend checking those out. As well as microwell-based platforms. So this is one also from the Greenleaf lab using the Takara isolate system. We've actually been using this now as well with the combinatorial indexing strategy that I'll talk about in a minute, as well as some work with the Cellsea platform that I'll talk about right at the end. So the main platform we use in the lab, though, is this single-cell combinatorial indexing strategy. And the big advantages that we see with this platform is there's combinatorial scaling of throughput, so it's really easy to scale up to much larger numbers of cells at a low cost. So it's a nonlinear cost expansion based on the number of cells that you're profiling. So if you profile more cells, it'll be a lot cheaper per cell. It doesn't require any specialized equipment. It's adaptable to a variety of properties, as I'll touch on at the end, and it's very inexpensive. The most expensive portion is doing PCR. So the way this technique works is we start out with whatever cell type we want to have, whether it's culture or tissue. It works on fresh or frozen. Isolate nuclei. And one advantage here compared to RNA-seq strategies is when you're dissociating cells, they often have a lot of time to kind of change their transcriptome while they're undergoing this kind of radical change when they're being dissociated and are still alive and viable. So this kind of kills everything and locks it in place, which is kind of nice. We then distribute these cells, or nuclei, into one or more 96-well plates, where we just distribute them out. So we have somewhere between 500 to 6,000 or 8,000 nuclei in each well of the plate. And these nuclei are permeable so that we can then go in with a transposase complex and do the attack-seq library preparation. But instead of having just the regular universal sequencing adapters, we have adapters that have unique DNA barcodes that correspond to the well of the plate that the transposition is happening in. So the transposase enters in, acts on the chromatin. The transposase stays bound, so it kind of glues that library molecule in place. So now these nuclei each contain a barcoded ATAC-seq library. We can then pool all of them together. So if I did one 96-well plate and grabbed a random nucleus out of the pool, it'd have a random one of those 96 barcodes. If I reach in and grab a second nucleus, there's only a 1 in 96 chance it has the same barcode as the one that I just pulled out. So this is the birthday problem in statistics. And we leverage that low collision probability to distribute a limited number into each well of one or more new plates, such that it minimizes, and we have a fairly low probability of having any two nuclei with the same barcode. And then we do another round of barcoding with PCR indexes. So we can tell out, discriminate between nuclei with the same transposase barcode if they're in two different PCR indexed wells. So our resulting sequencing molecules have two sets of indexes. So we actually do 4 index read runs along with the read 1 and read 2 to get that barcode combination. And it's that unique combination that gives us the ability to profile many more cells relative to the number of wells that we were actually using in the assay. All right, so the first project I want to talk about is one on corticogenesis, where we do some improvements to it. This we put up as a bioRxiv preprint not too long ago, so you can check it out there if you want more details. I'm just going to talk about a couple aspects of it here. But the first part of this was an optimization of ATAC-seq. So one thing we found, if we did this ATAC-C assay, where you actually just use a fluorescent transposase, if we stain our nuclei with DAPI, so the blue, and then red for the transposase, what we found was that we get pretty limited penetration of the transposase into the nucleus. So, and that was concerning to us because we know there's a lot of active chromatin in the center of the nucleus. So we thought about ways that we could actually improve that and found this chemical compound marketed as pitstop2. And we did some optimization experiments and found 70 micromolars, the right concentration. What it does is it actually opens up the nuclear pores. So we get more transposase that can actually enter into the core of the nucleus. And we can quantify that from the imaging data, where if you look at the bold red line is using the pitstop2 reagent as we go into the core of the nucleus and the distance from the periphery, and the dashed yellow line is without using this reagent. So there's a marked increase. It's still not uniform like we would like, but it's certainly better than what we had before. And then this translates directly to increased unique reads per cell. So we're actually getting more transposition events. And we've done quite a few additional experiments in different cell types to show that it doesn't even affect the profile. It's just an increase in the signal that we get. And it ranges between some preps and some samples, it's maybe 2-fold improvement. Other samples, it might be an 8- or 9-fold improvement. And you can see this is the log 10 unique reads here with different concentrations of using the pitstop2. So we wanted to apply this to something. So we have a project going on in the lab. It's a collaboration with Brian O'Rourke. This is where we're taking these patient-derived induced pluripotent stem cells and using the spin omega technique to actually differentiate these into 4 brain cortical organoids. And just to show that they actually stain for the markers that we want, we got a lot of beautiful images. There's pages of these in the preprint if you want to check those out, like this, where there's lots of different marks, and just to show that we're getting the nice cortical layering that we would expect and the nice rosettes in these cortical organoids. We then took these at either 30, 60, or 90 days in vitro and performed our Sky ATAC-seq experiment, and then analyzed it for the chromatin accessibility patterns, either at the local or global level. And just to show that the pitstop2 reagent, we had 2 organoids that did not have pitstop2 and 1 that did. And again, this is the log 10 unique reads on the x-axis, and you can see there's a pretty substantial improvement. I think it ended up being an improvement of a median of about 3-fold in the unique reads per cell. We then take these matrices and put it into a tool called Systopic, which has been an excellent tool for improving our ability to analyze single-cell ATAC-seq data. It really pulls out a lot of structure that we weren't able to resolve before, and then visualize the data using UMAP. So here's a projection into 2 dimensions of these cells. I think this is something like 12,000 cells, so a relatively small data set compared to what I'll show a little later. But and if we look at things like organoid identity, it's fairly uniform, at least with respect to days in vitro. So we can see that the earlier 30 days in vitro start out at this little nub here, and then the 90 kind of span almost everything throughout the whole landscape. And we also noted that the pit stop 2 treatment doesn't really have an effect on where the cells distribute in this space, as well as in higher dimensional space too. So the first thing we did was identify these clusters. This is using just Louvain-based clustering, and identified these 5 individual groups. So 1 and 2 are neural progenitor cells, 3 are outer radial glia, 4 are these intermediate progenitors that are shifting and progressing into full neurons in cluster 5. So we can do differential accessibility analysis and identify sets of peaks exclusive to each one of these individual clusters, and then do a motif enrichment analysis. And this, with all the literature that's available in the corticogenesis space, it was pretty easy to identify exactly what these were based on different marks. So like different SOX genes and NeuroD, for instance, and the later cluster 5, where there's a pretty high enrichment for those. And so that's looking at kind of global landscape enriched peaks within clusters, but we can also look at the local level. So in these plots, I'll show a couple later as well. If you look at a region of the genome where each one of the lines across here is a single cell, and the points are the signal, you can see PAC6, which should be active just in the early neural progenitors. We've got a lot of signal, same with SOX2. And then that signal kind of decreases as we move into the later, more neuron-like clusters. EOMs kind of increases more in the middle and end, and then genes that should really only be active in the more late glutamatergic neurons have the most chromatin accessibility around these distal elements proximal to the genes. So the other thing we can do is pseudotemporal ordering. So all these were derived from original iPSCs. This is using monocle 3. And it does look kind of messy. That's because we did it all in 3 dimensions, but you can't plot or project things in paper in 3 dimensions. So we had to reduce it all to 2 dimensions. So that's why it looks kind of tangled here. But it's actually a pretty nice progression going through here. We have 3 different branches in the neuronal lineages going from this progression here. And what's also really interesting is we have topics. So these are sets of peaks that come out of that systopic tool that are associated with these individual branches. So it actually gives us sets of loci that we can really target in to see and compare between the branches to see what they're actually doing and what these branches are. It was hard to resolve them before, but we've generated quite a bit more data in this project, and we're able to kind of tease out what these individual neuronal lineages look to be. So we can also look through pseudotemporal ordering at the global motif accessibility for any given motif in the genome. So we take our database of motifs and see how accessible those loci are with respect to the population and kind of z-score it. So this is using something similar to what the ChromeVar tool is. So we can see the progression of increased accessibility for certain neuronal marks later on, but not early on. We can then take it to the local scale, and there's a tool called Cicero that's also from the Cole Troutnell's lab. And what this does is it correlates regulatory elements, the signal between them across single cells, and enables us to actually link these regulatory elements to one another. And these links correspond and correlate very well with things like Hi-C data or other ways of actually physically measuring the association of chromatin within the nucleus. And as I mentioned before, the promoter accessibility doesn't correlate very well with gene transcription, but the signal at linked distal regulatory elements actually does correlate well. So when we use this tool to link our distal regulatory elements to individual promoters, we can aggregate that signal and use it as kind of a proxy for gene transcription. So we get these gene activity scores that we can then use in a way that one would use RNA-seq. And that ends up looking like this for the transcription factors, which is very similar to what we would expect based on known literature and corticogenesis, where we see increases in neuron-related genes actually a little earlier than we end up seeing the motif increase, and same for a variety of different marks. So we can then put these two together, where we can take so shown in solid here is the motif accessibility for these transcription factors. And then the dashed line is the gene activity score used by measuring the distal regulatory elements around that gene. So it's kind of combining this local gene activity from these individual genes with the resulting effect of these genes becoming active, producing the transcription factor, then acts on the chromatin and opens up those loci. So it's pairing those two. And we use this to dissect out the rest locus. So we actually couldn't get the gene activity score for PAX6 because the promoter kind of overlaps with other promoters, so you can't resolve those. But for the other genes, we were able to, and particularly in REST here, which is, we love REST because it's got this nice, big 23 base pair unique motif that's really easy to distinguish. And it acts to kind of slow the progression of neurogenesis. So in the beginning, it's not really needed, so the gene activity score is low, the gene isn't active, and the motifs in the genome aren't open and accessible. But then as we progress through, as we get to these outer radial glia, where REST is needed to slow that progression into the neuron state, the gene activity score increases, and then the resulting effect of that gene activity on the global chromatin increases the accessibility of the motif, and then it decreases as we get to these intermediate progenitors where the gene activity score goes down, and then after that, the motif accessibility disappears, and the cells progress into full neurons. We're excited about the ability to kind of pair these two local and global effects together. So next, another neuroscience-based story that I wanted to talk about is trying to actually incorporate spatial information into this single-cell ATAC-seq assay. So for RNA, like I said, you have this nice poly-A tail. Also, RNA is in the cytoplasm, so you can use strategies like spatial transcriptomics, where you've got an array with barcodes corresponding to positions, and use that to access your RNA. For ATAC-seq, all of the molecules are in the nucleus, which is hard to access in that type of format. So you can't use those same strategies, but we thought, well, we can do high-density multi-regional sampling, which has been used for a while, and actually just increase the resolution more and more until we get something that's biologically meaningful. So what we do is we take, in this case, a mouse brain and take coronal slices, and we alternate between thick and thin slices. So the thin ones go off to imaging or cyclic immunofluorescence, and then the thick ones get laid out, and we do these microbiopsy punches. I should note that this figure here is very much not to scale. This was like an old proof of principle diagram that I submitted for a grant. But the actual size, if you look at some of the numbers there, the punches are more along the size of like the circle inside the 8 or 9 is more the size of the punches that we're actually doing. So it's much higher resolution than what this diagram shows. But what we can do is take these punches, dissociate the nuclei, and one advantage of our combinatorial indexing assay is we've got all of these barcoded transposase complexes that we can use for sample multiplexing at that stage. So each one of these microbiopsy punches can get one or more individual transposase barcodes, and we can track them all the way through. So there's actually quite a bit of optimization that had to go into the plate-based nuclei isolation from these microbiopsy punches, and due to the efforts of Casey in my lab, we were able to actually get very high yields of nuclei from these individual punches. Typically, we do 250 micron diameter punches because that's the smallest that you can buy, but we've started fabricating 100 micron punches, and those we were able to extract enough nuclei to use the assay for those as well. So that's the resolution that we're really targeting. And then just to show you that the imaging works as well, here's one of the 20 micron adjacent slice to a 100 micron thick slice, and we're able to image it for different marks. We also found that decreasing the diameter of the punch biopsy increases our yield, which kind of makes sense. If you've got a smaller piece of tissue, it's going to dissociate a little better. So what I'm plotting here is the punch volume in cubic microns, and whether it's a 200 or 300 micron section thickness, and whether we're using a 250 or 500 micron diameter punch. And you can see the ones in black that are the circles filled in are the 250 micron punch, and the y-axis is the efficiency, essentially, how many nuclei per cubic micron perform a lot better, which is why we've really shifted to smaller and smaller punches. So just to show you one of these experiments in particular that we've done, we took a set of actually 6 slices from the region around the somatosensory cortex, and we took 6 punches per slice. So 2 punches were in the upper cortical layers, 2 punches in the lower cortical layers, and then 2 punches from regions of white matter where we don't expect there to be neurons. We do our plate-based nuclei isolation, and we actually have enough nuclei that we can dilute that out into 4 separate plates. So each one of those microbiopsy punches gets 4 individual transposase barcodes that we can then pool everything together, and we just did 2 PCR plates. This enabled us to get around 90 QC passing cells per punch. And again, this is a pretty small scale experiment. It's pretty easy to scale up the number of PCR plates, is how we typically expand things out. And just to show you what the data look like after taking it through Systopic and then into UMAP, we've got almost 8,000 cells where we have at least 1,000 unique nuclear reads that are in peaks per cell. Though I think the median unique reads per cell was something like 30,000 around there, so a pretty decent quality data set. And if we, instead of color coding by the 96 punches, because then it just looks like confetti, I color coded by either lower layer punches, upper layer punches, or these other punches from white matter. And what you can immediately see is a lot of these clusters have all 3 of the categories, but one of them generally only has 2. And as I mentioned, you know, the one that it doesn't have is other, which is white matter, where you don't expect there to be neurons. So even without any information about what these cells are, just the positional information, we can tell that this cluster here is definitely the neuronal cluster. We've done quite a few of these preps, and one thing that's kind of nice about single-cell ATAC-seq data is that there's not much of a batch effect, certainly not compared to RNA. So even without doing any batch effect normalization, we can take whole brain data, a prep where we did 384 punches, and then these 3 different categories and combine them all together, do Systopic and UMAP. You get a pretty even distribution of the different samples across the different cell clusters, with the exception of one cluster here that was only in whole brain, but it was a region that we didn't punch, so that kind of makes sense. So if we go back to this data set I was showing before, we can then take these data and color code by the transcription factor motif accessibilities. So each cell is color coded by that, where red is increased accessibility. And you can see this one cluster here for SOX9 is really high, which is a marker for astrocytes, so that jumps out really nicely. And then as I mentioned before, the cluster in the upper right are neuronal clusters, and they have higher accessibility for transcription factors related to neurons. So that makes sense. We can also take things to the local level to look at marker genes. So C1q, A, or C is a marker gene for microglia, and we see a lot of accessibility around the promoter and proximal regulatory elements for C1q, AC for the microglial cluster. We can do the same thing for oligodendrocytes for MOBP, which is a marker gene for that, where you get a lot of accessibility around the MOBP gene and associated regulatory elements, as well as a cluster here. A bunch of these microglia cells have a lot of accessibility for a region here that we're not sure what that is, so that was kind of an unexpected novel finding when we were plotting this gene, so we're digging into that now. And using those strategies, kind of combining the global motif accessibility with these local marker gene accessibilities, we can pretty readily identify what each of these clusters are and what the cell types are that are present. So in theory, we can do these plots with respect to every single one of the clusters, but it's a little easier to just visualize just splitting things between neuronal versus non-neuronal cell types. And we can do things where we take the slice and we just mark them based on where the punches came from, whether it's upper, lower, or other, and the proportions of each of the cell types, or whether they're neuronal or non-neuronal. So as you would expect, the other cluster only has around 3% neuronal, whereas these upper and lower have about 50-50, which is around what we would expect. Or we can actually project it through these different slices and have the proportions of neurons for upper and lower, where again it kind of hovers around 50%. There's not much variation with respect to this axis going through the somatosensory cortex, or for other, except this little peak up here, which could just be punch position. We actually have each of these punches recorded and imaged. But then the last thing we can do, and what we're particularly excited about, is the ability to do, I guess, pseudo-spatial ordering. We're doing ordering using monocle based on the epigenetic profile, but then we can overlay our spatial information. So in this toy case, which is just really a demonstration of the technology, we already know what markers are there for upper layer neurons and lower layer neurons, so there's not a lot of new information. But let's say we didn't know that, and there's a tissue type that hasn't been studied as extensively as the cortex. we can use this and actually get this progression and then overlay our spatial information and know, okay, now we know that all the markers associated with these cells up at the top here are associated with upper layer neurons. So it enables us to get that information. And we're also starting to apply this to a disease model, so a stroke model where we can induce an ischemic event in one side of a mouse's brain. And then you get this radiating effect out from the injury site of astrocyte and microglial reactivity as well as neuronal deterioration and death. And we can actually punch out, radiating out from the injury site and see the effects on the epigenome extending out from the injury site. So that's the next application that we're pretty excited about. And one other note that I think if anyone out here works with anyone or does any research on banked brain tissue, it can be quite difficult to work with, and unfortunately, fortunately we have a brain bank at OHSU where we've got 4,000 or 5,000 brains. Unfortunately, a lot of those are preserved this way, where you take the tissue from an autopsy, and we take a pretty large piece of that, and pieces that aren't specifically dissected out for projects just end up getting placed in a -80, which means they're freezing very slowly, which is terrible for tissue preservation. And it's very frustrating that there's like thousands of samples that are preserved that way. And then you could wait 3 to 5 years or so as the samples that we've been testing. But all of the work that we put into isolating these nuclei from the spatial preps, we came up with a pretty optimized protocol for isolating brain nuclei. And it actually enabled us to work with these samples that were previously not accessible to us. The tissue would just be too broken down. And this is just an example. We can then stain those nuclei for nu-N, a marker for neurons, and we get a nice split between those two cell types, whether they're neuronal or non-neuronal. And then we can perform our SkyDNA assay, and this is just showing the y-axis is log 10 unique reads. Each point is a single cell. The bottom kind of region is just noise. And we also compared it to mouse flash-frozen brain tissue, which is preserved in kind of an ideal way, and the library preparation actually looked very similar. So you can see kind of the blue and salmon-colored distributions at the top are very similar. And then actually taking that through to UMAP and cystopic and UMAP, we get the respective clusters that we would expect. And I should note we were targeting 80% non-neuronal cell enrichment, and we got 80.3, so very, very close to what we were hoping for. So we were quite excited about that. So last I wanted to talk about some future directions. So these are kind of very new areas in the lab that we've been working on. So as I had mentioned before, a lot of the other strategies that people use for single-cell attack-seq involve this physical compartmentalization, whereas we use this barcoding schematic. Well, we reasoned that we could probably increase the throughput of our barcoding schematic by incorporating some of these platforms. I should note, Jason Buenrostro had a paper in Nature Biotech where he did a great job doing droplet-based combinatorial indexing. So I just want to note that. It's a really excellent paper. We tried to get droplet-based single-cell attack-seq working for about 3 years, and actually he did as well. And it was just such a pain. In theory, it should work pretty easily, but in practice, droplets tend to fuse really easily when you're trying to do this attack-seq chemistry. So it took a lot of work, and we ended up giving up after about 2.5 years, and reasoned that microwells don't fuse, right? And it was just issues with oil, apparently, with the droplet-based strategy. So it happened when we were moving away from droplets. This Cellsea platform came out. It's a microwell-based platform. They designed it for single-cell RNA-seq. We're using it a little bit for that, but primarily single-cell attack-seq. So the way this technique works is we isolate our nuclei and do our in situ transposition, so our transposers can enter in, act on the chromatin. We then gravity feed the nuclei into these microwells. They're about 30 or so microns in diameter in kind of a honeycomb shape, a hexagon shape. We then gravity feed in beads, and you can actually fit a single bead into each well, and you can actually occupy almost every single well with a bead. And then do rounds of linear amplification or PCR that actually have used the barcodes in the bead to amplify these fragments. So this is very similar to the chemistry that goes on in the droplet-based strategies from 10x or Bio-Rad. And then we get our read with a bead index for the microwell. So the way this typically works is that it's Poisson loading, so we add in typically around 10% of the number of nuclei of the number of microwells on the chip. So if we have a 100,000 microwell chip, we'll load 10,000 cells or nuclei, whereas a million, we'd load 100,000 nuclei. So when we first started working with this, there were some challenges. The first is bead quality. So anyone who's been doing single-cell strategies, the quality of oligos on a bead is typically pretty poor, and that has had a huge impact on our assay. So that's been one big struggle that we've finally been able to sort of work through. Also getting nice nuclear lysis, because as I mentioned, the transposase kind of stays and glues that library molecule in place. So releasing that has been a bit of a challenge. And then also thermocycling on the instrument with an oil that doesn't evaporate has been a challenge. So we spent all that time dealing with oil for a droplet-based strategy. And then we moved to this platform to get away from oil because we were so sick of dealing with all of that. And then the first thing we have to troubleshoot is oil. So go figure. But some of the early experiments before we got the oil worked out actually started working pretty quickly, where we can show, here's a region of the genome where we've got some nice peaks. There's a fair amount of background, but it's not too bad. If we zoom in on one peak, you can see that you've got nice ataxic signal, and we're able to really readily call peaks. So we're pretty excited about this. We're now doing lots of additional iterations of experiments to improve the quality of these, but it's pretty close to where we're targeting. But of course, our goal wasn't just to do regular microwell index single-cell ATAC-seq. We want to use combinatorial indexing as well. So this schematic is pretty similar, where instead we do our plate-based and barcoded transposition. We then distribute these nuclei per well by gravity, except instead of one nucleus per microwell, we can increase that number and add in our barcoded bead and do the same linear amplification, but now the resulting molecule has the transposase index as well as the bead index. And it looks more like this, where instead of having around a 10% utilization of the microwells, we're able to get effectively 100% utilization of microwells, and you can add in multiple nuclei per microwell. In practice, we've been able to load comfortably around 10 nuclei in each microwell, and then also add in the bead. We haven't taken those through prep and sequencing because the scale is actually quite high, where it's still Poisson loading, but you have a really high value for lambda, where let's say you target 4 nuclei per well. If you're using a 1 million microwell chip, that's a 4 million cell prep. So I haven't been able to make slides from the data because it just came off the sequencer, I think Thursday or Friday, using a very similar strategy to this, but we have our first prep where we've produced almost 2 million cells from one single prep. And then the challenges there become actual software tools to handle that number of cells. Certainly anything R-based, if we've started running into this challenge where R has this maximum of around 3 billion non-zero entries in any given vector, and we routinely exceed that, even from data sets far smaller than this. So we're kind of struggling with ways to get around that and other tools, because just the scale of this is quite large. And so kicking off an analysis on even a 1 million cell data set, you can often run a tool, and a few months later it might not be finished. So please, if you're going to work on improving anything, we would really be happy about anything that would increase the speed of doing analysis on very large cell data sets. So as I touched on at the beginning, one thing we like about combinatorial indexing is it's pretty versatile and can be applied to a lot of different properties. I've just been talking about this one in the top left for ATAC-seq for chromatin accessibility. Here's several other publications. This is through collaboration of my group with Jay Shendray, Cole Trapnell, and Illumina, where we've been working together for several years now in terms of extending and adapting this single-cell combinatorial indexing technology. One I'd like to focus on just a little bit is the SkyDNA-seq approach. So this is a way of getting single-cell whole genome sequencing, where, and this is published, so I'm not going to go into too much detail in Nature Methods, but instead of targeting just open chromatin, we actually found a way to remove nucleosomes from the intact nucleus and open up all of the DNA. So now the transposase can actually insert everywhere in the genome to give us single-cell whole genome sequencing. And we applied this to several things, but I think the most interesting little vignette we had in the paper was a primary stage 3 pancreatic ductal adenocarcinoma, where we performed SkyDNA-seq, we did subclone identification, and actually we're able to identify 3 distinct subclones based on copy number profile, looking at breakpoint windows. And what we found later that's not in the publication is we can actually aggregate cells across these and then do single nucleotide variant calling. This has also been improved in a preprint that's out, that's a collaboration with Jay Shendre's lab that's really been led by Yi Yin, who's a postdoc in his lab, where we incorporate the Leonti strategy. So this is incorporating a linear amplification with in vitro transcription to amplify the genomic DNA signal. And that's been working well, where it can actually get about a 20 to 25-fold improvement in the number of unique reads per cell compared to this original SkyDNA-seq prep. We've also extended it to single-cell DNA methylation and an unpublished component where we're able to actually access regulatory sequences in addition to DNA methylation in the same cells. So the methylation portion is published, where we just had some adaptations. It's bisulfite-based. And one of the stories we had was showing that we could apply this to primary mouse brain tissue, where we can split out different cell populations based on their methylome, and identify inhibitory and excitatory neurons as well as non-neuronal populations from just the methylome. But we've been able to extend this now to using the principles in GnomeSeq, where we can take a methyltransferase that methylates at GC dinucleotides. So this is different from the typical nucleotide context that methylation generally occurs in, which is CG. And we can artificially methylate regions of open chromatin. So the same effect of steric hindrance of enzymes not being able to work on closed chromatin applies here. So it'll methylate these open regions in this detectable context. So then when we compare the 2 different methylation signals based on their context, we can discriminate between the 2. So if we just anchor a window at CTCF sites in the genome, and you look at the blue pattern here, you can see a marked decrease in DNA methylation around the CTCF binding site, which makes sense. CTCF is a methylation-sensitive binding protein. And then if we look at the GC methylation percentage, so this is a measure of the chromatin accessibility, we get a spiking signal here, which is the nucleosome positioning, which is, CTCF does a good job at actually anchoring where those nucleosome positions go. And then we get this big increase in accessibility and then a dip in the middle. And what's pretty cool is that this dip here is actually the footprint of the CTCF protein on the DNA. And also I should note, this technique, we haven't done a whole lot with it because it's just extremely expensive to do the sequencing. Cost-wise, even though the library prep is extremely cheap, the sequencing side, this is about a little over 100-fold more expensive per cell when compared to single-cell ATAC-seq. So just kind of a disclaimer not to run out and try doing single-cell methylation because it is quite costly. But as I mentioned, we're planning to increase throughput, but our goal is to take those strategies that I've just been talking about and merging them with our combinatorial indexing microwell-based platform to enable million cell data sets from lots of different properties. Probably not methylation because of the cost, but for a lot of these other properties, including some strategies we have for implementing it on single-cell Hi-C, as well as single-cell chip-seq type strategies. So that's the direction we're going in with these approaches. And with that, I want to acknowledge the people that were involved in these projects, people in my lab, as well as OHSU, some collaborators at Cellsea, certainly my funding, and collaborators at other institutes, and certainly my lab, who actually did all of the work. I haven't touched a pipette in about 5 years, and unfortunately these days I don't even do much more analysis, so it's really them that get all this done. So with that, I'll take any questions. Thank you.